In February 1865, a quiet Augustinian monk stood before a small scientific society in the city of Brunn in the Austrian Empire. He was not famous. He did not belong to one of Europe's great universities, and the subject of his lecture did not sound revolutionary. For years, he explained, he had been growing peas in a monastery garden. He had crossed tall plants with short plants, yellow seeds with green ones, smooth peas with wrinkled ones. Then he had counted the offspring, generation after generation, looking for a pattern hidden inside life itself. The men in the room listened politely. A few may have understood parts of the mathematics. Most did not realize they were hearing the foundation of modern genetics. Mendel's paper was published the following year, circulated in a small regional journal, and then almost disappeared. He died before the world understood what he had done. Yet today, every discussion of inherited disease, DNA testing, selective breeding, genetic engineering, and even the characteristics you received from your parents begins in some way in that forgotten garden. And at the center of it all was one of the most ordinary foods on Earth, the pea. But peas were feeding human beings thousands of years before they helped explain heredity. Their story begins near the birth of agriculture itself, in the lands stretching from the eastern Mediterranean through the Near East. Wild pea relatives grew among grasses and rocky hillsides. Their pods were small, their seeds were hard, and when fully ripe, the pods could split open and scatter the peas before anyone could harvest them. For wild plants, that was a successful survival strategy. For hungry humans, it was a problem. The first farmers slowly changed that. They saved seeds from plants whose pods stayed closed longer, whose peas were larger, and whose harvest was easier to gather. Nobody knew the language of genes or mutations. Nobody drew Punnett squares. But every time a farmer chose one plant over another, human hands pushed the pea in a new direction. Over generations, the wild plant became a dependable crop. Archaeological and genetic evidence places peas among the ancient group of crops domesticated in the Near East, alongside early wheat, barley, lentils, and chickpeas. These early peas were not the bright green, sweet vegetables found in freezer bags today. They were usually left on the plant until the seeds dried. Dry peas could be stored for months, carried over long distances, and boiled into thick soups or porridges. In a world where one failed harvest could mean starvation, that mattered more than flavor. Grain provided energy, but pulses, such as peas, provided valuable protein and made poor diets more complete. Even the plant itself helped farmers in a way they could not yet see. Bacteria living around the roots of legumes captured nitrogen from the air, improving the fertility of the soil and helping future crops grow. Peas followed farming communities into Europe, North Africa, and parts of Asia. They appeared in the food traditions of ancient Greece and Rome, where they were eaten in soups, stews, and simple meals. They were not normally symbols of wealth. They were practical food, compact, storable, and difficult to ruin. Soldiers could carry them, sailors could store them, families could keep them through winter. Long before peas became associated with neat green rows on a dinner plate, they were survival food. That role would become even more important during the Middle Ages. Wheat and other grains could fail because of rain, frost, war, or disease. Bread was culturally important, but bread depended on a chain of labor. Fields had to be harvested, grain had to be threshed and milled, and flour had to be baked. Dry peas were less glamorous, but they were reliable. Across Europe, peas appeared in thick pottages eaten by ordinary people. The old rhyme about peas porridge hot remembered a dish that could sit in a pot, be reheated, and feed a household again and again. For centuries, the word peas referred to the food as a mass, almost like porridge or grain. Eventually, people began treating the final sound as if it meant a plural, and a single seed became a pea. The language changed as the crop changed. Medieval pea fields were generally mature and dry, but in the early modern period, European growers increasingly selected varieties that tasted pleasant when eaten young and fresh. This created a completely different experience: tender green peas that had to be picked at exactly the right moment. Fresh peas became fashionable because they were difficult. A dry pea could wait in storage. A fresh pea could not. Once picked, its sugars quickly began turning into starch, and the sweet flavor faded. Serving delicate green peas therefore required gardens close to the kitchen, laborers ready to harvest them, and cooks prepared to use them immediately. At elite European courts, especially in France, fresh petits pois became a seasonal luxury. Their appeal was not only taste, they demonstrated access, timing, land, servants, and control over nature. At the court of Louis XIV, the fashion for young peas became so intense that writers described nobles eating them late into the night and speaking about them as if they were jewels. Madame de Maintenon described the obsession as both a fashion and a madness. The peas were cooked with butter, herbs, lettuce, and sometimes cream. What had been once a hard seed kept against hunger was now being harvested immature and served as a delicacy. The difference was not a new species. It was human selection, timing, and status. People had transformed not only the plant, but the meaning of the plant. The pea had traveled from the food of prehistoric farmers to the tables of aristocrats. But its greatest transformation would happen far from a royal dining room, behind the walls of an Augustinian monastery. Gregor Johann Mendel was born in 1822 to a farming family in Moravia. He understood plants not only as scientific objects, but as living things tied to work, weather, and survival. After joining the Augustinian order in Brünn, he studied mathematics and natural science in Vienna. That combination would prove decisive. Many plant breeders before him had crossed varieties and noticed that offspring resembled their parents. Mendel approached the problem differently. He wanted numbers. The pea was almost perfectly suited to his plan. It grew quickly. It produced many offspring. Its flowers normally fertilized themselves, allowing a plant to preserve a characteristic across generations. But Mendel could also open a flower, remove the pollen-producing parts, and introduce pollen from a different plant by hand. Most importantly, pea varieties displayed clear, contrasting characteristics. Plants could be tall or short. Seeds could be round or wrinkled. They could be yellow or green. Flowers could be purple or white. These were differences he could classify without guesswork. Before beginning his main experiments, Mendel obtained dozens of pea varieties and spent time determining which ones bred true. A true breeding tall plant produced tall offspring generation after generation. A true breeding short plant did the same. Only after establishing stable lines did he begin crossing them. This patience was essential. If the starting plants were not predictable, the results would be meaningless. Imagine Mendel working in the garden. Each flower had to be handled carefully. Unwanted self-pollination had to be prevented. Crosses had to be labeled. Seeds from each plant had to remain separate. The next generation had to be planted, grown, classified, and counted. A single careless mixture could damage years of work. For roughly 8 years, Mendel studied thousands upon thousands of plants. The first generation seemed simple. When he crossed a true breeding round plant with one wrinkled, the offspring were round. The wrinkled characteristic appeared to vanish. The same thing happened with other pairs. Tall seemed to defeat short. Purple flowers seemed to defeat white. Yellow seeds seemed to defeat green. A less patient observer might have concluded that one characteristic had destroyed the other. Mendel did not. He allowed the first-generation hybrids to reproduce. Then the missing characteristics returned. Among the next generation, the dominant-looking characteristic appeared in roughly 3 plants for every one showing the hidden characteristic. Round peas produced wrinkled descendants. Tall plants produced short descendants. The characteristic had not been erased. It had passed through the first generation unseen. This was the clue. Take seed shape as an example. A plant carrying two round versions produced round seeds. A plant carrying two wrinkled versions produced wrinkled seeds. But a hybrid carrying one of each still looked round because the round version was dominant. When two hybrids reproduced, their offspring received different combinations. One could inherit two round versions, two could inherit one round and one wrinkled, and one could inherit two wrinkled. Visually, that produced the approximate 3-to-1 pattern Mendel kept seeing. At the time, many people imagined inheritance as a kind of blending. A tall parent and a short parent should produce something permanently medium-sized, just as black and white paint create gray. But Mendel's peas did not behave like paint. Their characteristics remained separate. One could be masked and then reappear intact. Mendel proposed that inherited characteristics were controlled by paired factors, one received from each parent. Today, we call those factors genes, although modern genetics is far more complex than the simple model. Each parent carried two versions of a factor but passed only one into a reproductive cell. When offspring formed, the pair was restored. This explained why a recessive characteristic could disappear and then return. It also explained Mendel's famous ratios. The pattern was not magic, and it was not random chaos. It was probability. Mendel then studied more than one characteristic at the same time. Seed color could pass independently of seed shape. A plant did not have to inherit every visible feature as one package. Different herited refactors could be sorted into new combinations. This principle, later called independent assortment, helped explain how offspring could resemble their parents while still being unique. The achievement was extraordinary, not because nobody had ever grown hybrids, but because Mendel turned breeding into an experiment that could be measured. He controlled the parents, he followed multiple generations, he counted large samples, and he used mathematics to reveal a biological law. In a garden full of vines and flowers, he recognized that life was preserving information in discrete units, and almost nobody cared. Mendel presented his findings in 1865 and published them in 1866 under a title usually translated as Experiments on Plant Hybrids. The paper did not become famous. It appeared in the Proceedings of a local natural history society, a publication with limited reach. Some scientists received copies, but the work did not transform biology during Mendel's lifetime. The scientific world was discussing evolution, variation, and the origin of species. Yet the mechanism of inheritance remained deeply uncertain. Part of the problem was timing. Mendel's mathematical approach was unusual for many botanists. His conclusions were buried inside a detailed paper about plant crosses. The word gene did not yet exist. Chromosomes had not been understood as carriers of inheritance. DNA had not been identified as hereditary material. Mendel had discovered a pattern without being able to show the physical object behind it. He continued working, became abbot of the monastery in 1868, and found himself consumed by administrative responsibilities. He experimented with other plants, kept bees, and recorded weather, but he never achieved recognition equal to the importance of his pea work. When he died in 1884, he was respected locally as a clergyman and administrator, not celebrated worldwide as the founder of a new science. Then, around 1900, three European botanists, Hugo de Vries, Karl Correns, and Erich von Schermach, encountered similar inheritance patterns and brought renewed attention to Mendel's forgotten paper. Suddenly, the old pea experiments looked prophetic. Scientists began connecting Mendel's factors with chromosomes. William Bateson became one of the strongest defenders of Mendelian ideas and helped popularize the new field. In a letter written in 1905, Bateson introduced a name for the science of heredity: genetics. Recognition did not end every argument. Scientists debated how widely Mendel's laws applied, whether his numbers were unusually close to expectation, and how his simple factors related to the messy variations seen in nature. Those debates continued for decades, but they did not erase the importance of his method. Mendel had demonstrated that biological inheritance could be tested quantitatively. Biology was no longer limited to describing organisms. It could predict outcomes. The pea patch had returned from scientific obscurity. Over the following decades, Mendel's principles joined with chromosome research, evolutionary theory, and later molecular biology. Scientists discovered that DNA carries hereditary information. They learned how DNA is copied, how genes influence proteins, and how mutations create variation. Mendel did not know any of that. His laws also did not explain every characteristic. Many characteristics are influenced by multiple genes. Some genes are linked together. Environment matters. Dominance can be incomplete. Yet his central insight that inheritance depends on discrete units passed between generations survived. Even the wrinkled pea eventually gave up its molecular secret. In 1990, researchers showed that the wrinkled seed characteristic Mendel studied is associated with a disruption in a gene involved in starch production. Round seeds convert more sugar into starch. Wrinkled seeds retain more sugar and lose water differently as they mature, causing them to collapse and wrinkle. More than a century after Mendel counted those seeds by hand, molecular genetics connected the visible wrinkle to a specific change inside the plant. While peas were transforming science, industry was transforming peas. For most of history, peas had been eaten dried because fresh peas spoiled quickly. Canning changed that. During the 19th century, improved canning methods allowed peas to be sealed, heated, transported, and stored for long periods. Factories could process huge harvests near farming regions. Peas were among the earliest vegetables to enter the commercial canning industry. But early canned peas had a problem. Heat softened them and dulled their bright color. Some producers used coloring treatments to make preserved vegetables appear greener, practices that eventually became subject to stricter food regulation. Consumers wanted peas that looked and tasted as if they had just left the garden. The answer would come from cold rather than heat. In the early 20th century, Clarence Burnside developed and commercialized rapid freezing techniques after observing how food exposed to intense Arctic cold could freeze quickly and retain its quality. Slow freezing forms large ice crystals that damage cells. Faster freezing produces smaller crystals and preserves texture more effectively. Peas were ideal for the new industry, but speed remained everything. Once harvested, their sweetness begins to decline. Modern processors therefore shell, blanch, chill, and freeze peas soon after picking. The frozen pea became one of the great symbols of modern convenience. A crop once available fresh for a brief season could now wait in a kitchen freezer. Unlike many foods damaged by industrial processing, frozen peas preserved much of what people valued: color, sweetness, and nutrition. The vegetable that had once advertised aristocratic access became ordinary enough for almost anyone to keep at home. Peas also became machines' food. Mechanical viners separated peas from their pods at enormous speed. Plant breeders developed varieties that matured more uniformly, allowing entire fields to be harvested together. Agricultural scientists selected for sweetness, disease resistance, yield, size, and the ability to survive processing. Every improvement depended on the principles Mendel had uncovered with earlier pea varieties. Today, peas appear in forms their earliest farmers could never have imagined. They are sold fresh, frozen, canned, split, dried, powdered, and concentrated into protein. Pea protein now appears in meat alternatives, sports nutrition, snacks, pasta, and dairy-free products. The plant also matters because it is a legume. In partnership with bacteria around its roots, it can obtain much of its nitrogen through biological fixation. This can reduce dependence on synthetic nitrogen fertilizer and make peas useful in crop rotations. Farmers can grow peas before cereals, interrupt certain disease cycles, add diversity to fields, and support soil fertility. In a century increasingly concerned with climate, fertilizer costs, and sustainable protein, the ancient crop is being asked to solve new problems. Modern researchers continue studying pea genetics to develop varieties that tolerate drought, heat, disease, and unstable seasons. Seed banks preserve old varieties and wild relatives because characteristics discarded by past farming may become essential in the future. A wild pea with poor flavor might carry resistance to a disease. An old landrace with low yield might survive extreme heat. Genetic diversity is insurance, stored inside seeds, and that creates a strange symmetry in the pea's history. The first farmers changed peas without understanding genes. Mendel used peas to reveal how inheritance works. Modern breeders now use that knowledge to change peas more precisely. The crop taught us the rules, and we are using those rules to redesign the crop. The consequences extend far beyond agriculture. Mendel's ratios became the starting point for understanding inherited conditions in human families. They helped scientists predict how some disorders pass from parents to children. Genetics later transformed medicine, criminal investigation, ancestry research, conservation, and biotechnology. None of these fields can be reduced to a single monastery experiment, but the intellectual doorway Mendel opened remains visible in all of them. A pea looks insignificant. It is small enough to roll beneath a plate and disappear. It has no dramatic shape, no powerful smell, no obvious connection to empires or machines. Yet peas were present near the beginning of farming. They helped sustain ancient communities, medieval households, soldiers, laborers, and the poor. Fresh peas became symbols of luxury. Dried peas fought hunger. Frozen peas helped build the modern food industry. And in one monastery garden, peas answered a question humanity had asked for as long as parents had looked at their children. How does life pass its characteristics forward? It was hidden in purple flowers, wrinkled seeds, and plants that grew either tall or short. It was hidden in numbers that repeated across generations. Mendel saw the pattern, but the world was not ready to see it with him. 35 years after his work was published, science finally returned to the garden. The humble pea had done more than feed civilization. It had shown civilization how inheritance works. Not bad for something most people push around the edge of a dinner plate. If this story changed the way you look at an ordinary pea, subscribe for more hidden histories behind the foods that shaped the world. And tell us in the comments which everyday food we should uncover next.